Hi, everyone. We're going to give just a minute or two for people to start trickling in. We have a lot to cover today and an exciting topic, 2 trials to talk about. So we will get underway here in just a moment. All right. I can't wait. I'm getting started. All right. Welcome, everyone. And thank you for joining us on the 14th episode of Ewing's U. I'm Emily McFadden, and I'll be your co-host today. I'm one of the co-founders of Little Warrior Foundation, and more importantly, a godmother to a now 13-year-old warrior named Lucy. Ewing's U is a production of the Little Warrior Foundation. We're a national cancer foundation with a laser focus on finding a lasting and permanent cure for Ewing sarcoma. We were founded in April of 2020, and now we've granted out over $1.6 million in high potential therapies and clinical trials for increasing survivorship and reducing toxicity. We appreciate all of your support so that we can continue our mission of bulldozing our way to more effective, less toxic therapies. At Little Warrior, we are obsessed with science, and Ewing's U is our way of bringing the leading sarcoma experts and cutting-edge ideas to the Ewing's community. Today, we'll be hearing from one of the prominent experts in this field, Dr. Federico. Specifically today, she'll be exploring 2 unique trials offered at St. Jude. A little bit about Dr. Federico. After her education at Stanford, Virginia Commonwealth, and then back to Stanford, Dr. Federico joined St. Jude as a fellow in 2007. Today, she is a faculty member in the Division of Solid Malignancies. And as a clinical scientist, Dr. Federico is passionate about advancing the cure rate for patients with high-risk neuroblastoma and sarcomas by identifying, testing, and optimizing novel therapeutic strategies. As a clinical trialist, she has led numerous trials as the principal investigator. We'll be hearing about a few of those today. And next year, she will be opening an international phase 3 trial for newly diagnosed high-risk neuroblastoma patients. In addition to the care and research she conducts at St. Jude's, Dr. Federico serves on many important national and international committees for the advancement of solid tumor care. Her commitment to advancing treatment has been recognized by countless awards, and we are so honored to have her here today. Before we kick things off with Dr. Federico, I wanted to encourage you to post questions throughout the webinar using the Q&A function on Zoom. My lovely co-host, Hillary, will help facilitate the questions after the presentation. Hillary, would you like to introduce yourself?
Sure. Hi, everyone. I'm Hillary Lang. I am blessed to be a warrior mom to Angel Ian Lang, and he's my he's my guide in all of this, and he got me into all of this with his diagnosis of Ewing sarcoma in 2018. He did pass away in 2021, and since I have put all of my efforts into finding a cure for Ewing sarcoma or advancing anything I can do. And I'm also a geek with science, and got to meet with Dr. Federico a while back, and we thought that it would be really great to have her here. So thank you so much for being here.
So Hillary, yes, as a trenches trained oncologist, she is a really valuable member of the Little Warrior team. She's on the scientific review team. So as ideas and proposals come through, Hillary is right there in the mix of it all. And I'm especially grateful to have her here today as Piero, who is our primary science guy, is tending to his little warrior. So very grateful to have Hillary and Dr. Federico with us today. And with that, I will pass things off to Dr. Federico. You can share your screen and take it away.
All right, sounds great. Thank you so much for having me here with you all today. Let me put this on the big screen. Is it in the right mode? Yeah.
There we go. Perfect.
All right. Well, today I thought that the primary focus is going to be primarily on one trial that I've worked with, and then I'm going to present a little bit on a different trial that one of my colleagues is working on. And really the goal here is targeting vulnerabilities in Ewing sarcoma cells. And so, of course, you all know a lot about Ewing sarcoma, and really what has inspired all of this work is just what Emily was saying and just what Hillary were saying, that we have to do better. So patients who have in the bottom right-hand corner, you can see that patients who initially have metastatic disease, many of them are unfortunately not able to be cured long-term. And then if their cancer has come back, for all patients, especially those who relapse less than 2 years out from the completion of therapy, we have got to do better for these patients. And so that's really what has inspired a lot of this work. And so an obvious treatment strategy for our patients would certainly be to try and target that fusion protein, the EWS fly, or it oftentimes has or can have other partners. But unfortunately, lack of enzymatic activity and disordered structure have made that really difficult to target. And so some of the work that I'm sharing today is really trying to attack Ewing sarcoma in a different way. And it was based off of these couple of papers that came out a few years ago in the mid-teens, which essentially showed that Ewing sarcoma cancer cells were sensitive to DNA damage. And could we then try and target this with a class of drugs called PARP inhibitors? And so this is some work that was done by a colleague of mine at St. Jude. She's an excellent scientist named Elizabeth Stewart. And what Dr. Stewart did in the mid-teens is she was also looking at this issue with DNA repair damage. And so in the top row, you can see that that's a cell line U2OS. So that's an osteosarcoma cell line, most common bone tumor in pediatrics. And then the bottom row is a Ewing sarcoma, the Ewing sarcoma cell line, ES8. And at time 0, where the lightning bolt is, these cells were exposed to radiation. And really, in this case, radiation was meant to damage the DNA of those cells. And you can see that at 5 minutes, both the Ewing sarcoma cells on the top and the osteosarcoma cells on the top and the Ewing sarcoma cells on the bottom inquire damage. That's evidenced by the pink. And then over time, the osteosarcoma cells, they're able to repair that damage. And so at 2 hours, 8 hours, and by 24 hours, they're all back to blue cells, healthy cells. But the Ewing sarcoma cells just continue to incur damage over time. And so this was kind of an aha moment to a lot of people and really made us think, can we target this DNA repair defect like the researchers in the breast cancer, BRCA mutated breast and ovarian cancer target these cells? And so the way I like to think about this is, if I have a piece of paper and I tear the piece of paper, that piece of paper now has damage, but I have a toolbox. And so I can use tape, I can use staples, I can use glue to put that tear back together. But if I only had one tool and then you took away that tool, such as staples, and that's the only tool I have, then now that is going to continue to be a damaged paper. So this is kind of a similar idea, that the cancer cells themselves are not, Ewing sarcoma cells are not able to repair. They don't have all the different tools. And so because they have issues with something called homologous recombination, if you block one of their other tools, then when the cells are damaged, then hopefully they cannot repair themselves. So that really set up the subsequent preclinical studies and clinical trials. And so this class of drugs called PARP inhibitors, then the next question was, well, there are a whole bunch of PARP inhibitors, and this is all work that was done in a mouse model of Ewing sarcoma. And so is there a specific PARP inhibitor that is best, and what medicine should we combine the PARP inhibitors with? And so Dr. Stewart, in a mouse model, looked at these different questions. And so here, the red box surrounds the single agent PARP inhibitors. So just to orient you for this graph, basically on the y-axis, that is how many are alive, so mice. So 1 equals 100%. And then on the x-axis, it's days and time. And you can see that if you gave these mice that have Ewing sarcoma single agent PARP inhibitor, it doesn't work. And all of those mice have progressive disease, and it is not effective. when Dr. Stewart combined the different PARP inhibitors with temozolomide, so a drug that's continually used in patients with Ewing sarcoma, then she did see some response. And so here, just to orient you, the blue line is a PARP inhibitor called veliparib. The red line is a PARP inhibitor called olaparib. And the green line here is labeled as BMN 673, but it's now called talazoparib. And the black line, what the black line is in comparison to is irinotecan temozolomide. So, we oftentimes will give our patients irinotecan or temozolomide if their Ewing sarcoma has come back. And so this really served as a comparator. What does a PARP inhibitor plus temozolomide, how does that work in the mice compared to standard second-line therapy that we use in our patients? And so again, the green line here, talazoparib plus temozolomide, had about 60% of the mice that were cured of Ewing sarcoma. She then combined the different PARP inhibitors with irinotecan, same situation. You can see here, though, that the drug olaparib, as well as talazoparib with irinotecan in combination, were successful at getting rid of curing some of the mice with Ewing sarcoma. But definitely in her preclinical studies in a Ewing sarcoma mouse model, the agents that worked the best was the PARP inhibitor plus irinotecan and temozolomide. So really, I'll refer to that as the triplet drug combination with the three of them. And so what does that look like here? Looking at it a little bit differently, this preclinical work. So again, just to orient you, these are mice that have Ewing sarcoma that has been grown in the bone marrow space. And so it's come out of the bone, similar to the microenvironment that Ewing sarcoma is in our patients. And here, the green line are those mice that received the talazoparib plus irinotecan and temozolomide. And you can see that all of those mice have these bright areas go away and they all develop a complete response, CR, complete response. And this is in comparison to the irinotecan and temozolomide that was administered to the mice, in which all of those mice developed progressive disease. And so again, this is just looking at, these are called xenogens, where you have the irinotecan temozolomide after many cycles. The tumor does not go away, and after the completion of the therapy, you can see that the tumor below it in a petri dish. Single agent talazoparib doesn't work, but again, in the murine model, in the mouse model, the triplet drug combination was effective. And so everyone was very excited about these findings, and that led to a clinical trial called BMN. And so to give you kind of an idea of start to finish with that trial, Dr. Stewart's preclinical studies ended in April of 2014. So that's the left-hand side of the slide. And at that point in time, the Children's Oncology Group was actually taking a clinical trial forward with talazoparib and temozolomide. And so we reached out to them to share the findings, to just say, hey, the triple drug combination looks like it's best in this murine model, but that trial was so far along in development that they weren't able to add it. And so it went on to study talazoparib plus temozolomide. So then we started calling our collaborators to see if we could get pharma on board. The talazoparib at that time was owned by a company called BioMarin. And so when we presented Dr. Stewart's data to BioMarin in June of 2014, they said submit a concept. And then they very quickly approved the concept and then approved the protocol in actually 3 months, which is very remarkable timing, because a lot of times these things take quite a bit longer. So in this case, actually the study opened and the first patient was enrolled on this clinical trial called BMN in under a year, right under a year from when the preclinical trials initially finished. And so that was exciting to be able to get the trial to patients, and we were hopeful that this would be the cure. Unfortunately, as drugs often do, they change hands. And so Medivation actually bought talazoparib midway through the trial, and they were going to actually discontinue the trial. But again, with a lot of advocacy from our part and others, we were able to keep the trial open. And then actually Pfizer acquired Medivation. And so again, we had to go through the whole rigmarole of, can we please keep this trial open? here's the preclinical data, our patients need it, and we were able to get them to allow us to continue keeping the trial open. And then we reached accrual on this phase 1 study, dose finding study, in January of 2019. And so what did BMN look like? It was a phase 1 study open to children and young adults and adolescents. As many phase 1 studies in our pediatric population, it was open actually to all patients who have solid tumors that are outside the CNS. And it was okay if patients had received prior irinotecan or temozolomide exposure. And so the first arm was just arm A, looking at the double drug combination, the talazoparib, which is taken by mouth, and irinotecan, which was given in the IV formulation. But the goal was always to get to the triplet drug combination based on the preclinical work. And so once we achieved the doses on arm A, then we were able to look at what the dose of temozolomide that the Children's Oncology Group had been able to achieve with talazoparib on their developmental therapeutic study, and then determine the dose levels to start at for the triplet drug combination. I should say though, and I think this is important, going into this study, Dr. Stewart, what I didn't show you is she did a lot of PK studies, pharmacokinetic studies, where the mice that had the Ewing sarcoma tumors were exposed to the drugs, and then she tried to determine the concentrations that were achieved in those mice, and then try to compare those concentrations to drug concentrations that we will oftentimes see in our pediatric patient population. So this is called a mouse equivalent dose. It's not perfect, right? But it's the best that we can do. And we knew going into this study that we were probably going to in order to see the same results that she saw in the mice, we were probably going to have to kind of hit the top doses of the triplet drug combination. And indeed, that's, I think, what really happened. And so thankfully on this study, there were some patients who had some treatment responses. And so here are some examples of some pulmonary lung mets, pre-treatment on the left, post-treatment on the right, in which the tumors did get smaller for some patients and nearly go away. But we all know that it wasn't the cure that we were looking for, and it certainly isn't what we saw in the mice and in the mouse model. And so we saw this kind of signal of activity in Ewing sarcoma and a few other solid tumors, but we were really limited by this issue with toxicity. And so the patients, many of whom come with lots of therapy, prior therapy, and kind of exhausted bone marrow because of all the things that people like me have done to it. We're having a lot of hematologic toxicities with their counts being down. So in this case, platelets and neutrophils, and then also some GI toxicities related to their irinotecan, such as some diarrhea, poor weight gain, et cetera. And so the question then was, okay, can we mitigate these toxicities, still get these drugs to the tumor, and try and get around some of the issues that we were having with the systemic circulation of the drug? And so the solution was to change the formulation of irinotecan. And so there's a formulation of irinotecan that's been used in the adult patient population called ONITT. It's liposomal irinotecan. And what ONITT really is a liposomal formulation of irinotecan. And so you can see there are these green, I always talk with my hands, there are these green lipid bilayer, which really shelters the blue irinotecan. It allows the drug to clear and then get inside the tumor without having as many systemic side effects. It has a longer half-life than irinotecan, and it has lower plasma clearance. And so what happens is in circulation, about 95% of the irinotecan actually stays in that lipid bilayer. Once the liposomal irinotecan gets taken up into the tumor cells, then there are some macrophages that are inside the tumor cells that engulf the liposome and basically release the irinotecan, which is converted then to SN38. And that's actually the active part of the drug. And so we were hopeful that by changing out the irinotecan and using a liposomal irinotecan, that we could deliver higher doses of SN38 directly to the tumor, and that when in combination with the PARP inhibitors, that might lead to improved tumor response. And so back to the lab, we went, and this is it's just such a fun part of my job where I get to collaborate with my friends. And so it's great because as this trial was going on, I was having these conversations with Dr. Stewart, and she was able to start doing some of these mouse studies while the clinical trial was going on to try and decrease time delays. And so she did a number of different preclinical studies, again, in a Ewing sarcoma mouse model. And so here you can see that in this case, the onivide, that's the liposomal irinotecan, is labeled NAL-irino, but that's the onivide. And so when she exposed the mice to the onivide, the NAL-irino, you can see that there was this prolonged exposure and concentration in the bloodstream. That's evidenced by the red line over time versus standard irinotecan, which gets in and then gets out. And so then when she actually looked at inside the tumor, again, this all in mice, what happens with the mice and what happens with the tumor. And so you can see that at time 0, the mice receive the NAL-irinotecan, the onivide. You get to pretty high tumor concentrations fairly quickly by 24 hours. And then it remains in the tumor for about 4 days before it starts to go down. And versus in the plasma where it really goes down very quickly. And so again, we were hopeful that this would mitigate some of the toxicities that we saw initially, that inhibited dose escalation of the irinotecan. And so she performed some mice studies similar to what she did before. And so again, tumor is that right hind leg represented by the rainbow. And you can see that in this case, when the mice received the double combination, talazoparib plus the onivide, as evidenced by the red line, that those mice all had developed a complete response, a CR. And this is in comparison to the talazoparib with just standard irinotecan, in which some mice had a PR, but eventually they had progressive disease. And in comparison to standard second-line therapy with irinotecan temozolomide, that black dash line, which the mice all developed progressive disease. And so again, when she looked at the combination, this is a group of mice looking at placebo, which is just placebo, and the black line, irinotecan temozolomide, the black dash line. And then, actually my head's on top of this, I can't. Then the talazoparib and NAL-irinotecan is in the red, and the talazoparib NAL-irinotecan temozolomide is in the green. And you can see that the double regimen with the tal onivide was just as effective as the triplet before. And so that really set up the basis for the next study. Because we were going to be opening this phase 1 study, and we always accept all comers, solid tumor all comers, I thought it was important for us to also make sure that this was beneficial to different solid tumors. So indeed, onivide is a combination that can be useful in patients, or excuse me, in mice with neuroblastoma. I thought I took all these out, I'm sorry. And then also in rhabdomyosarcoma as well. So that's a question that sometimes we'll get when people are saying, but you're putting all these different diagnoses on a trial, is that okay? And in this case, this is an active agent against other cancers as well. And so how do we improve outcome for Ewing sarcoma? And so again, this is building on the fact that the double drug combination here, the talazoparib plus the onivide, actually led to a durable complete response in the murine model. So that really has set up the next study, which is called onivide. And so these studies, just to give people, again, a timeline, because I think sometimes it's like this black box, like how do you get from preclinical work to a clinical trial? So these studies were completed by Dr. Stewart in January of 2019. And then soon after, our BMN trial closed. Dr. Stewart and I at ASCO of 2019 really pitched this study to the 2 different drug companies who owned both the onivide and the talazoparib. Onivide is owned by a company called Ipsen. Talazoparib still is owned by Pfizer. And it's sometimes very difficult to get drug companies to allow their drugs in the same treatment arm, but with a lot of discussion and then also, again, relying on the preclinical data, we were all able to get into agreement. The concept was approved and the trial went forward. It took a long time for contracts, to be perfectly honest, but it did open in May of 2021. Certainly the pandemic didn't help any of these situations as well. So what is ONITT? Well, it is a phase 1/2 study that is open for pediatric solid tumors and Ewing sarcoma. And so in the phase 1, which is outlined by the red box, that's the phase we're currently at. Patients are randomized to receive either arm A, which is Onivide plus talazoparib, or arm B, which is Onivide plus temozolomide. And again, this is because irinotecan and temozolomide are frequently used in the second line or third line setting for patients who have Ewing sarcoma. Potentially getting higher concentrations of irinotecan with Onivide into the tumors might benefit these patients. So that is why the comparison is Onivide plus temozolomide and not irinotecan and temozolomide. The goal of the phase 1 study is, of course, to assess for tolerability and toxicities to try and determine what the recommended phase 2 dose is of these two medications are. Once we get to the recommended phase 2 doses, then on the right hand of the slide, you can see that we will perform a randomized phase 2 study in patients with relapsed or refractory Ewing sarcoma. And again, patients will be randomized to receive either Onivide plus talazoparib or Onivide plus temozolomide. At the same time that the phase 2 study is ongoing, there will also be some expansion cohorts in other types of solid tumors. So the primary objective, again, of the phase 1 study is to determine the recommended phase 2 doses of those two different combinations in patients with refractory or recurrent solid tumors. And then there are some secondary objectives, which are, of course, to describe the safety profile, to actually look at the pharmacokinetics, so what happens with drug concentrations in the patient's bodies after they've received the medications, and then really to look for response. I know I always spend a lot of time with my patients, really making sure that everyone understands the difference between a phase 1 and a phase 2 study. Of course, we always hope that patients' tumors will get smaller and go away. But really, the purpose of the phase 1 is to determine the doses with the hope that it will work. The eligibility criteria I included just because I didn't know if that might be interesting to people. And so it's pretty standard eligibility criteria for a phase 1 study. So children 12 months to under 30 years. Patients have to, for the phase 1 study, have relapsed or refractory solid tumors that do not have CNS brain tumors present. If you have a tumor in your skull that's maybe pushing in a little bit towards the brain, that is allowed because that is actually a bone met and not a primary CNS met. The patients, of course, need to meet a bunch of different criteria, which are kind of standard organ function criteria to make sure that when we give therapy that their organs are in good shape to receive the medicines and to have fully recovered from the other effects of different therapies. The trial is being conducted as a St. Jude investigator-initiated study, and I'm the PI, but then there are a bunch of external sites across the US and as well as Canada. And so the sites, not all of them are open yet. It's taken some time to get them open, but now we have 6 sites that are open, actually 7 if you include us, with kind of a really nice geographic distribution. And so I'm hopeful that more patients will be able to enroll and stay closer to their homes than having to travel to me in the middle of the country. And so again, in this phase 1 study, it's just a dose escalation study where we're trying to determine the right doses. It has been helpful to have the 2 different arms because oftentimes when patients are waiting to enroll in a phase 1 study, the trial might be closed for a period of time, but having the 2 different treatment arms has really allowed us to stay open. But it is important to know that the patients for the phase 1 study are not allowed to select the treatment arm. They are actually randomized. And that was not a choice by me, but a request of the FDA. And so that's what we're doing. This is kind of what the different treatment arms look like. And so anytime patients are enrolling onto studies, I think it's always, of course, important to have a sense like, where are you at starting dose wise, right? Because sometimes you might be enrolling on a phase 1 study and the initial doses might be so low that we really don't think that there's going to be activity, but you're kind of at a spot that you have to start there. In this case, the starting doses, so those are the stars next to the 1, dose level 1 is the starting dose, are actually really good doses. So the starting dose of talazoparib is an adult starting dose of talazoparib. And in the temozolomide arm, 100 milligrams per meter squared is a pretty standard dose of temozolomide that we give. And then the starting doses for Onivide, so again, dose level 1, 120 milligrams per meter squared, are actually very good doses. And so just to give you a sense of what that means, in the adult patient population where they have used Onivide, many of those studies, the dose is 70 milligrams per meter squared given every 2 weeks, 2 to 3 weeks. 70 to 90 really depends on the clinical trial or the indication. And so in this case, we were starting at a higher dose than in the adult studies. And the reason being is that there is a clinical trial, the results haven't been published, but there is a clinical trial out of the SPOT consortium looking at the combination of Onivide plus cyclophosphamide for patients with relapsed or refractory solid tumors. And again, the data is not out there, but in conversations with investigators and then in conversations with the drug company, we knew that they were at much, much higher doses of the Onivide, so felt comfortable starting at that dose level. It's also important here to point out that the Onivide in the initial trial was administered on days 1 and 8, and this was really to try and mimic a protracted course of irinotecan. And so there's a lot of data in the pediatric world that sometimes, patients receive irinotecan on days 1 through 5, and then the following week they get 5 more days on days 8 through 12. And so this was really to try and mirror that and have longer exposure of the tumor to the Onivide. And so the patients receive these courses, that's what the C stands for, every 21 to 28 days. It really depends. The trial is written as 21-day courses, but for some of our patients, as they've gone on, they've really fallen into more of a 28-day strategy. And initially the patients have disease evaluations performed after cycles 2, 4, and 6, but then it's spaced out after that. It happens after every 4 cycles thereafter. And they're allowed to stay on study and receive the therapy on study for up to 2 years. I think it is important here, at least it was important to me as I developed this clinical trial, that I wanted to make sure that our patients would have the opportunity to have radiation or surgery while on the clinical trial, since that is oftentimes not allowed on these clinical studies. And so on this clinical trial, it does make our stats a little bit trickier, but I think it's better for patients. And so on this clinical trial, then the patients are allowed to have radiation or surgery at any time point after cycle 4. And so as we went along, we realized that actually this dose schedule of giving Onivide on days 1 and 8 was not tolerable. And so we saw more than we would anticipate issues with GI toxicities especially. And so what we did as we started to see these is talked with my friend, Dr. Stewart, asked her to go back to the lab. You can see on the right hand of the slide, what she did then is she repeated some of the studies using that mouse model and getting rid of the day 8 Onivide to make sure that we would still see responses if we got rid of the day 8 Onivide, and she did in the mouse model. And so then the trial was amended, and we got rid of, with those little X's on the top, that day 8 Onivide. And since we have omitted the day 8 Onivide, the patients have tolerated the combination much, much, much better. So there are very, very few serious adverse events, and certainly not these dose-limiting toxicities that we were seeing with our original schedule giving Onivide on days 1 and day 8. And so, as I mentioned, once we get to the end of the phase 1, then, and again, patients with Ewing sarcoma, and indeed many of our patients who have enrolled in this study have Ewing sarcoma, but the phase 2 study will be focused just on patients with Ewing sarcoma. The patients who are randomized will be randomized on the phase 2 study. They will be stratified based on if their disease is refractory, meaning it has never gone away or has recurred less than 2 years, versus if it's recurred later than 2 years. And it will be okay for patients who have received prior irinotecan therapy, because we know irinotecan is being included in some of the upfront settings. The goal of this study will really be to look at the progression-free survival. So the time duration to relapse, and hoping that it's obviously, I hope no one relapses, but if they do, that it's significantly delayed. And then we'll look at the comparison between the two. And then there are a whole host of other secondary and exploratory objectives that we will look at as well. And again, on the phase 2 portion of the study, the patients will be allowed to have radiation or surgery after cycle 4 for local control. And so where we are at with that study is that it's accruing very well. Again, there was a pause when we had to make the adjustment in the clinical trial to get rid of that day 8 Onivide. And so that was a pause, letting the FDA and everybody else get on board with it. And then once we were able, so that pause was about 6 months of not being able to accrue patients. But now we're back up and running. We had to start over with our number of patients, since the schedule is different, but it's been accruing really well. We definitely are seeing some objective responses to therapy. I would love to show you this, but I can't because the clinical trial is ongoing, and then I would get in a lot of trouble. And our patient, the longest treated patient on this clinical trial, actually just completed 2 years of therapy and has had to come off study, because you're only allowed to receive it for 2 years on the study, but has elected to continue it off the clinical trial. And then there are a lot of patients who are approaching that 1 year on therapy. And so now I'm going to switch gears in the last couple of minutes and just briefly touch upon another clinical trial that we have, or even another therapeutic option. And so this therapeutic option is really focused on angiogenesis. And so angiogenesis is just the blood vessels that form in a tumor. And it's really important and necessary for the tumor to grow, to spread, to survive, and to persist. And the truth is, when you look at tumor vasculature, and it's very dysregulated. And so, normal structures will have kind of this nice highway of blood vessels. But when you look at tumors, it's just kind of a mess, where there are all these different little blood vessels, and they're squiggly, very tortuous, and there are many of them. And the bottom line is they're really dysfunctional and leaky. And so that makes it hard as a physician to get drugs uniformly to a tumor if the blood vessels are kind of a mess. And so the key regulators of blood vessel formation, angiogenesis, are VEGF, and PDGF, and then there are a whole bunch of others. And there are a lot of different drugs that can target these different regulators of angiogenesis. So here I'm just going to talk about a prior study that was done, because I do get called on this by physicians and by patient families sometimes. So this was a trial that has already been completed. It was called ANGIO-1. And what it was is a single institution phase 1 study looking at a 3-drug combination using sorafenib, bevacizumab, and then low-dose cyclophosphamide. So I know you all are very familiar with higher doses of cyclophosphamide that you get during initial therapy. And in this case, the cyclophosphamide is much, much lower dose. The idea is it's called metronomic chemo, where you're just taking a small oral dose continually exposing the tumor to that drug. It was, again, a phase 1 study, so really looking at dose-limiting toxicities and the maximum tolerated doses, and was conducted in patients with relapse or refractory solid tumors. And so the bottom line is, in this kind of gamish of a lot of different patient diagnoses, is that the therapy was very well tolerated. And so this is a toxicity table where, to orient you, we have these different grading systems of toxicity. Grades 3 and 4 are the more significant toxicities that we're really trying to avoid frequently. And there were very few of those across the board with this therapy. And then importantly, there was a signal of activity seen in this small patient cohort, with some patients, 5 of 17, having a response, and then 5 additional patients having actually stable disease. And so this is what that schedule looks like, is on day 1 of a cycle, which is every 3 weeks, the patient gets an IV dose of bevacizumab. This is usually done in an outpatient setting. And then they start taking by mouth cyclophosphamide daily and sorafenib twice a day, and just stay on that cyclophosphamide and sorafenib until they come back for their bevacizumab, in which case they just keep taking it. Importantly, the regimen is so well tolerated that it doesn't tend to drop counts to the point of requiring blood transfusions, neutropenia, which leads to hospitalizations. It's really well tolerated. So most patients just come, they get their bevacizumab, they pick up their oral prescriptions, and then usually you're not going to have to go back to the doctor. And so for quality of life, our patients have always said it's pretty amazing for their quality of life. So this was the expansion cohort on Angio-1. And again, small patient numbers. I have highlighted here or boxed in the red, the Ewing sarcoma, abbreviated there as ES. There were only 3 patients, but you can see that some of these patients were able to maintain at least stable disease with a very good quality of life for a long period of time. And indeed, it actually allowed some of the patients who were waiting to enroll in a different clinical trial, it allowed their disease to not grow during that time while they were waiting to go on a different clinical trial. This was then, since that expansion cohort stopped, for many of our patients who unfortunately have failed a lot of different prior therapies, this is definitely a regimen that we sometimes offer them, especially when patients come to us really requesting, I don't want to be in the hospital a lot. I don't want to come to this clinic a lot. I really want to focus on quality of life. And so here, this was a recent review. I don't know where all it looks like my references went away, but I can send those later. I'm sorry. Where we looked at patients who had been treated with this regimen over the past few years. And so at our center, that was 14 patients with Ewing sarcoma. And then you can see that for the most part, the medications are very well tolerated, with a few things that I would like to point out. So one of the things that can happen with sorafenib is something called palmar-plantar erythrodysesthesia, which is where you get this, it's kind of like, it can be a painful rash. What we know about this, though, is that even if someone says, I'm starting to feel a bump or it's starting to hurt, a lot of times if you go to the twice daily dosing down to just once a day, typically resolves. And then we also start some other medicines. Other things that are always really important to watch out for with this treatment regimen, though, is that the patients can develop proteinuria, where they're excreting protein, and can have high blood pressure, as with other TKIs. And so we always have to watch for this. And then for patients who have big, bulky disease in their lungs, there is a risk sometimes that especially as tumors respond, that sometimes you can get a little hole called a pneumothorax that happens in their lung. And so anytime families are considering this regimen, I always just want to make sure that they're kind of aware of these other risks that aren't typical with other chemotherapeutics. And so for that group of patients here, it's broken down, and this is called a swimmer's plot, where you can see on the top, those are all osseous sarcomas, so Ewing sarcoma and osteosarcoma. And again, a lot of patients, this is not the cure. I don't want to even pretend like it is. And indeed, a lot of patients actually, as their best response, have just had stable disease. But have been able to maintain a good quality of life with limited time coming to the hospital. And that's sometimes been a nice bridge while they're waiting to go on to another therapy. So this has certainly served actually as a building block for another trial called Angio-A, not to be confused with Angio-1. And so again, Angio-A is a trial that's really focused on the vascularity. And so again, really trying to go after the vascularity, the angiogenesis of the tumor cells. But the addition, where Angio-A is different from Angio-1, which I just presented, is that we've added, in the primary oncologist for this trial is a woman by the name of Jessica Gartrell, where she's then added another drug that's really trying to release the brakes on the immune system, a PD-L1 inhibitor called ozalidomide. And so the question there is, can we target both the vascularity as well as the tumor microenvironment? And so in this study that is recently opened, it's a phase 1 study. And if you look at the red box there, the patients initially get Angio-1 therapy. So again, that's the IV bevacizumab, the atezolizumab on day 1, and then they get daily sorafenib and cyclophosphamide, like the prior trial that I discussed. Initially, there's a safety run-in, kind of a feasibility phase to make sure that this is feasible in patients and that they're able to tolerate the addition of that fourth drug. And then there's an efficacy phase in which there are more patients that will be included in the efficacy phase. So this trial is certainly open to patients with Ewing sarcoma currently in the part 1 safety run-in phase. I should mention, and I clarified with Dr. Gartrell right before this call, that currently in the efficacy phase, in the phase or in the part 2 of the trial, there isn't specifically slots for patients with Ewing sarcoma. However, they are going back to our scientific advisory group at St. Jude and going to be adding that other solid tumors, which of course Ewing sarcoma tumors would fall under. But currently, because they're in part 1 of the phase 1, the safety run-in phase, patients with Ewing sarcoma are eligible to enroll. And so that includes patients who are less than 30 years of age. And I think I already said most of that. And so that kind of brings us to the end. So clinical trials really are a huge collaborative effort. They take a great team of people working together. And I'm certainly blessed to be at St. Jude, where we have a great team. And then, of course, we can't do anything without our patients. And so I'm immensely grateful to our patients who always allow or enroll in our clinical trials. And really, we're in this together to try and make advancements in cure rates. So with that, I'll take questions.
Great.
Thank you.
Thank you so much. That was fantastic, super informative. And I especially appreciate you kind of taking us through that bench to trial and just how long and difficult sometimes that process can be. So we had some questions come through, and if people have more questions, keep loading them through the Q&A functionality. Hillary, I'll let you take the first pose the first question.
Sure. So, well, I had one really quick one, just myself. There was that SPOC trial that they did use Onivide in. How come Dr. Stewart didn't try with that first, with cyclophosphamide first?
Yeah, so it's a great question. So some of it is the mechanism of action for, and I didn't show that some of that data, but when she looked at her mouse models in the mice, definitely the best combination was with irinotecan and temozolomide.
So she did test with the.
Okay. Yeah, and then the SPOC trial, I really hope that we're gonna know the results of that at some point.
Ian was actually on that for about,
I remember you saying
cycles. That. Yeah, 10 cycles, and he did excellent and was completely free of disease and went to transplant. So that's why I was like, well, they already have this information on cyclophosphamide. Why can't we just keep continuing, and then you could eliminate that whole section of the trial and move on to the triplet? But no.
Yeah. So part of it is that we don't have the data.
Yeah.
And I really, we need it.
We need it.
The community needs the data.
Okay. Will you consider a trial that includes all 3 drugs, since it showed up? Probably eventually you will. Since it showed the most efficacy. Also, I hope this trial has an age limit that is above 25.
Yeah, so I think talking about the ONITT study. And so the plan initially was just to do the 2-drug combination, because that was just as efficacious in the mice with Ewing sarcoma and compared to the 3-drug, and trying to also eliminate some of that toxicity. So the goal now is not to do a 3-drug, a triple drug combination, but of course that may have to be something that we resort to if it's not working the way we want it to. The point about the age limit is so important, especially for sarcoma patients, because obviously a lot of them are older. So this trial on it goes up to the age of 30. And that's pretty similar with a lot of COG trials as well. The trials that allow for a higher age range are like those that are conducted through SARC and other excellent consortia. And that's usually because they're in partnership with other adult facilities that they're able to do that. But this one only goes up to age 30.
Okay. How many patients enrolled and the goal?
Oh yeah. So I didn't even talk about that. So the patient enrollment goal for the phase 1 study is that it goes to 18 for each arm. And so when we first initially started the trial, and then we hit some of those toxicities with that day 8 Onivide dosing, we had enrolled 9 patients. And so of course, when we eliminated the day 8, then we had to start back over. And so we need to get to our new number has to be 45 because it includes the first 9, obviously, because we need 18 per treatment arm. And so we are in the mid 30s.
Okay.
So
and
going
target-
along.
If people are, oh, sorry, Hillary. If people
Go
are
ahead.
interested in that trial, how do they get more information or hear their?
Yeah. So yeah. And I'm happy to, I can also send you, Hillary and Emily, something that has contact information. I'm happy to do that as well. So any clinical trials that anybody is ever interested at St. Jude, there's a St. Jude referral email address. It's like all over the website. And so that's one way. You can always email me directly and I can get you that information. And then what we typically do is we have a solid tumor coordinator. His name is Joe Bergantz. He's like a mystical unicorn. He's amazing. And so he works with patients and their families and their physicians, because we definitely need communication with the physicians to be able to know what treatments you've received previously, to make sure that we get any of that information. Sometimes families just want to consult. Other times families want to know, hey, am I eligible for this trial? And then what do I do to get there? And so he's able then to work with the physicians and the families to get them that information.
Is there any thoughts of adding a PD-L1 inhibitor to the Onivide trial?
Yeah, so good question. There isn't at this point in time. And PD-L1 inhibitors, of course, it's added in that second trial that I was talking about. There's a lot of debate and no one really knows what to do in this space. And actually, even when you look at the publications that people have on PD-L1 expression on tumors, there's like variability depending on who you're asking. But no, our plan is not to add that at this point in time.
I had a quick question too. Say someone's doing awesome and they finish the trial like so many others have in the past, that 2-year span. Would you consider using the PARP inhibitor for maintenance?
Yeah, so it's a good question. I think PARP inhibitor as a single agent certainly in the mouse model doesn't make sense. And I think the reason being is that you need the chemotherapy to damage the DNA. And then what normally happens, you have 2 strands of DNA, you damage the DNA, you break one, and then you block PARP from fixing it. And so then the cancer cell eventually dies. And so for Ewing sarcoma, which is different than BRCA mutated cancer, you really need the chemo to damage the DNA. So I don't think single agent PARP inhibitor is probably going to do it. But you do bring up an important question, which is what do you do after? In one of our patients' cases who elected to continue to get on those medicines, because I think he was just nervous about stopping, is that both of those drugs are commercially available. We obviously knew what dose he needed because he'd been on the trial for a long time. And so he's able to receive those medications off therapy using the doses that he received on the study.
That's good. Okay.
Yeah.
I was worried about him.
Yeah, I know. I know.
The next one. Sorry, for ONITT, will patients who have had tumor growth on irinotecan temozolomide be eligible?
Yeah, so they are eligible, and they're actually eligible also if you've had prior irinotecan exposure in the phase 2 study. And the reason being is that if we had to exclude those patients, then most of them would be excluded. We also know that by we, I mean like pediatric oncologists in general, that sometimes patients who relapse, even if they've previously seen that drug, will sometimes respond again. And then on the BMNRN trial, the patients that we saw respond had actually seen prior irinotecan as well. And so again, I don't know if it's, and a lot of the patients that we're currently enrolling have had prior irinotecan and seeing some responses. So I don't know if it's just that the concentration of the active part of the drug, SN38, is higher, or if those patients would have re-responded to irinotecan. I'm not sure exactly why, but it is not an exclusion criteria. The only exclusion criteria for going on it in terms of drug combinations is if you've received a prior PARP inhibitor and irinotecan as a combination, then you're not allowed. Yeah.
That makes sense.
Yeah.
Target timing for phase 2?
Yeah. So, what is it? It's November 30th. I'm hoping that we will wrap the phase 1 maybe by the end of the first quarter next year, by like springtime. And then we need to look at some PK data and of course get like the green light from the FDA and all the company on all these green lights. They always take longer than I would like them to take. But then we should be able to open the phase 2. And so I mean, I imagine the phase 2 will be open next year. That is the goal.
That's awesome. I did have a question about the angio-1. So I really like the concept that they don't have to have the treatment that's very intense. And does it affect their lymphocytes, white blood cells? Like, is that a dose limiting thing with that?
Yeah, so angio-1, so again, that's the combination of bevacizumab and the IV, daily oral sorafenib at the low dose, and then daily or twice daily, depending on who you are. Excuse me, daily or twice daily sorafenib and daily cyclophosphamide. That does not tend to cause neutropenia.
That's like if someone needs a break, I feel like that is a really good option.
Yeah, and it's actually just what I recently did with one of my patients who was responding to therapy, but having a lot of toxicity, different therapy, having a lot of toxicity. And she wanted to continue to take some medicine with hopes that it would keep her cancer from growing, but be able to get back to school. Because every time she dropped her neutrophil, she'd end up in the hospital with fever and neutropenia, and to not require so many platelet transfusions. The only, every once in a while, for the patients who have had like their counts affected, are patients who have had like, I don't know, many back to back. And so sometimes with them, we will dose reduce the cyclophosphamide a little bit and it's enough. And then but the bigger issue is sometimes with their platelets. I don't know if people on the call use it, but we've, for patients who have issues with platelets, using romiplastim has really helped a lot of our patients keep their platelet count up and allow them to stay on therapy.
That's
All right,
awesome.
one final question before we wrap up for the angio trials, because they're, I mean, really focused on quality of life. Do people need to come to Memphis to participate in these trials, or can they do that from remotely?
Yeah, so for angio-A, they do have to, because that's the addition of the PDL1, and because that's in the dose finding phase. It is a clinical trial that's just open at St. Jude currently. But if you want to receive the angio-1 regimen, the bevacizumab, cyclophosphamide, and sorafenib, then no, 100%. Like, those were clinical trials that were done a long time ago. We know the doses. Like I said, I've oftentimes sent physicians, if they've had questions or people, just sent them the papers, because that's something that they can do.
Perfect.
Oh, there's one other thing I should mention. I know you said that's it, but there's another trial that might be interesting to the patients with Ewing sarcoma, and that's that we have a B7H3 CAR T study. As soon as I didn't even think about tossing that in here, and
It's
so if pardon.
Another whole episode.
Well, I'll give you the expert on that. His name's Chris Lorenzo and Rebecca Epperly. Anyway, I know that, CAR T cells obviously haven't busted everything open for our sarcomas yet, but I know a lot of times families are looking for those. So this is a trial that's open at St. Jude. If people have questions, they can, or are interested in it, of course, just same people that you can reach out to. What they do first is they actually request tumor tissue. It doesn't have to be your current tumor tissue if you've had something biopsied in the past. And then our pathologist looks at the B7H3 expression on the surface of the cancer cells. And if you have a high amount of that, because I think if you have low, it probably wouldn't work. So if you have a certain amount, then you might be eligible to enroll in that trial. So I didn't even think about tossing that in earlier. Sorry.
No,
No,
that's great. It's exciting to hear that there's
other options.
in a space where it's characterized by very limited options. So thank you. And thank you so much for all you do in your commitment to the kids and in the solid tumor space. So we are going to wrap things up. For everyone following in, be sure to subscribe to our emails so you get updates on all of the Ewing's U episodes. Previous versions, previous recordings are available on our blog. We will have this up in the coming days. And so in closing, we have a sign off here at Little Warrior that embodies our fight in honor and in memory of so many little warriors and big warriors who have faced this awful cancer. So it's how we sign off all of our emails and all of our communications. So 1, 2, 3, swords up. Thank you so much.
Thank you so
Everyone
much.
have a great day.
Bye.
Bye-bye.